Good morning, my dear students. Myself, Kausar Momin, 8th Standard, Subject Science, Chapter number 10, Cell and Cell Organelles. This is Science 2 part. It is related about biology and all. So this is very new chapter for us. We will go ahead with this chapter. Before to start this chapter, we will recall few points. Point number 1. You can observe here on the screen. How many types of cells are found in living organisms? So obviously about cells, if you count it, so if you won't able to count it down first of all, and its number is billion trillion, means it is around 10 raised to the power of 13. 10 raised to the power of 13. You can imagine on 13 zeros these many cells are present in the living organisms. Okay. Question number 2. Which instrument had you used to observe cells? Obviously electron microscope. Okay, under microscope only we can observe these cells. With the naked eye you cannot observe. Okay, so last year we studied that cell is a structural and functional unit of living organisms. According to the functions, cells of different sizes and structure are found in different organs. We have body organs. With every body organ, different cells found over there. Okay, and there are cells present in animals as well as living things like human being things. So few things are different and few are commons in the cells. Okay, so we will go ahead with the structure of the cell. Let's go ahead with the structure of the cell. Here we have to fill it out. Structure of the cell. These two diagrams are given over here, plant and animal cell. This is plant and animal cells, which we have to fill this name. So I will show you one diagram related the same plant and animal, which is a same diagram. You have to recognize the name of the plant cell and animal cell, and you have to write down in your textbook. Let's go ahead with the screen. Observe this screen. Here you can observe plant cell structure and animal cell structure. The names are also given over here. The names which are common in plant cell and animals, these are given over here. You can observe, like cell wall present in plant cell as well as animal cell. Cell membranes present in only plant cell. It is not present in animal cell. Okay. Next is ribosomes are present in animal cell as well as plant cell. Nucleus is present in animal and plant cell. Nucleus is present in animals and plant cell. Vacuole is also present in only plant cell. Cytoplasm is present in plant cell as well as animal cells. Why I am saying you, I will show you. So this is about your structure of your plant cell and animal cell. Please observe the names and write down in your textbook. In the textbook, names, we have to fill the names. Just please observe this screen and write down the names in your textbook. Okay, so the same diagram I have shown you with the name. Pause the video over there and keep the textbook front of you and write down the names of these things during my lecture itself. And after the lecture you have to draw this animal cell as well as plant cell in your notebook also, rough notebook or your fair notebook. It should be a science notebook. Okay, so this is a part of your homework also. Here you have to draw this animal cell and plant cell. Now we will go ahead with this component animal cell, plant cell, which I have shown you in the previous screen, that cell membranes is present in animal cell as well as plant cell. cell wall is present in animal cell as well as plant cell, that you have to recognize plastids where it is present, animal cell or plant cell, or in both. All these answers you will get in the diagram which I have shown you before this. Okay, so this is also a part of your homework. You have to complete this table. Now we will go ahead with the components of a cell. It is found around cells of large fungi and plants. Animal cells lack cell wall. Cell wall is an elastic and strong coat around cell membrane. It is mainly composed of carbohydrates like cellulose and pectin. Afterwards, polymers like lignin, suberin, cutin are added in cell walls as per need. Functions of cell wall are support and to protect the cell by preventing entry of excess water in the cell. Obviously, anything in our body, it has a protection. Suppose if our skin only, if the skin get harmed, then it is opening, and after a few days the opening gets closes automatically. Obviously for the skin also there is a membranes for the protection. Okay, so similar way inside the skin the cells present in it, and for the same there is a protection, there is a protect layer, and it will support for the cells present in it. Okay, and whatever the excess water present in the cell, it should not go away from the same. That is why that protection cover is there. Okay, next is plasma membranes, or which we can say it as cell membranes. It is thin, fragile and elastic, covering that separates the cell components from outer environment. Whatever the outer environment, it should not enter in it. That is why there is a covering. So here is the structure of cell wall. You can observe here structure of cell wall. This is the structure of cell wall. So this is middle lamella. You can observe here primary cell wall. This is plasma membrane, soluble proteins which are present in it, soluble microfibers which are present in it, and hemicellulose, and this red line is of pectin. So all these things, all these the parts of the structure of the cell. So we can say structure of plasma membranes. Protein molecules are embedded in two layers of phospholipids. Plasma membrane is said to be selectively permeable membrane as it allows some substances to enter the cell while prevents other substances. Due to this property, useful molecules of water, salt, oxygen enter the cell and carbon dioxide exits the cell. Obviously in our body, everywhere with every section carbon dioxide entry is there and exit is also there. For the same, there are pores over there, and through the pores it will exit. Okay, if any changes occur outside the cell, the cellular environment does not change due to plasma membranes. This condition is called as homeostasis. Means if the skin is getting harm, then whatever the harmness is there, it gets fulfilled. So outer side of the environment, it will not affect on it. Okay, now you can observe here structure of plasma membranes. Before this we have seen structure of cell wall. Okay, and you can observe here in the cell wall there is a plasma membranes, this plasma membranes. Now we are going ahead with the plasma membranes, the detailed structure of the plasma membranes. Okay, so you have to draw the structure of cell wall also. Whatever diagram I am telling you to draw, you have to draw. Okay, for your extra knowledge. Now next is structure of plasma membranes. You can observe here we have seen in structure of cell wall the plasma membranes, and inside in it what are the things present in it, that we have to see. You can observe here protein, lipid bilayer because 1 and 2 layer, and few chains are there. You can observe here chain, so those chains are there. So this is the structure of plasma membrane. You have to draw this diagram also. So activities that consume cellular energy. So one is endocytosis. What is the meaning of endocytosis? To gulp, or we can say it as engulf food and other substances from outer environment. Whatever the outer to take entry, whatever the outer environment to take a food from that. So that is, there is a we can say it as absorption, quickly absorption. To gulp means to quickly absorption food from the outside the environment. Okay, next is exocytosis. Exocytosis means whatever the excretion is there, to excrete waste materials out of the cell. Obviously in our body there is a waste material which exit from anus. Okay, but with the cell wall also, in the plasma membranes also, there is a excretion of waste material, and whatever the excretion is there, we are saying it as exocytosis. Okay, so process of those that don't consume cellular energy. First we have seen which consume the cellular energy. Obviously something is taking inside and throwing outside. Obviously some part of energy is there. Okay, now what is the processes don't consume the cellular energy? Very first is diffusion. Entry or exit of small molecules like oxygen, carbon dioxide. As I said to you, in our body every corner there is a going and coming of oxygen and carbon dioxide. Okay, next is osmosis, the traveling of water from a part with more water to a part with less water through selectively permeable membranes is osmosis. So whatever the traveling of water in our body, that is from more water to less water, this is what? Permeable membranes, which we can say it as osmosis, and it is physical process with three possibilities. Now you can observe here, these are the three possibilities of osmosis. You can observe here osmosis in plant cell as well as osmosis in animal cells. Here are two diagrams over here, one is with the plant cell, another one is animal cells. First of all we will go ahead with the plant cell. Now you can observe here, this is the cell structure is there, and H2O is coming outside. Here H2O is going and coming outside. Here H2O is going inside. Okay, so these are the three stages. Next is hypertonic, isotonic and hypotonic. You can observe here, when H2O is coming out, H2O is going out, H2O is coming out, H2O is going inside it. Okay, so there is a process for the same, and we can say it as osmosis means process of traveling of water high to low, and it is present in animal cell as well as plant cell. Next is isotonic solution. Medium outside and inside the cell has same proportion of water, which doesn't go in or out. Let me show you isotonic solution. Here you can observe isotonic solution. Here the level of water is equal, because whatever the going part is there, that much is only coming out. Okay, next is hypotonic solution. The cell has less water than outside medium, so water enters the cell. So here only entering of water is there, nothing is going to come out, because this cell has less water and it needs a water, that is why only water entered in, nothing is going to come out. Hypertonic solution, the cell has more water than the outside medium, so water comes out of the cell. Example, fruit pieces are kept in a thick saturated sugar solution. The water from fruit pieces enter the sugar solution. So, the fruit pieces shrink after a while. If plant cell or animal cell is kept in a hypertonic solution, water comes out of the cell by the process of exomosis, and there is a contraction of cytoplasm. This process is called as plasmolysis. What is the meaning of this? Because what is happening over there, already water is there inside the body, already water is there. Now it wants to excess water is there, and it is whatever the water is there, it is the water is coming out. And example is given over there, whatever the, if your fruit keeping in the contact of sugar, then whatever the water present in the fruit, it gets attracted towards the sugar solution. Okay, next is cytoplasm. Here for the cytoplasm, we have to see the onion peeling. I'll show you first of all the where we are observing onion peeling. This is electron microscope. You can observe here. Under observation of this electron microscope, when you are observing onion peeling, so you will get this type of structure. So you can observe here cells of onion peeling, cell wall, nucleus. This is also nucleus. Cytoplasm, the very spot, minute spot is there, that is cytoplasm. And this is vacuole, the space in it. Okay, so this is the onion peeling when you are observing in under this microscope. So cytoplasm is the fluid between plasma membranes and nucleus. It is moving sticky substance. Many cell organelles are suspended in cytoplasm. Cytoplasm is the medium for cellular chemical reaction. The part of cytoplasm other than organelles is the cytosol. Cytosol stores vital substances like amino acids, glucose, vitamins, etc. In animal cells, cytoplasm is more granular and dense, while plant cells is thin and mostly pushed to the periphery due to large central vacuole. Let me show you for onion peeling. Here I will show you how to observe onion peeling under microscope. So first of all, we are taking over here onion. This is pure water, saffron mixed water, and that is another way is also a pure water. First of all, we peel the onion, and we are taking a very thin layer which is present in the onion. We are taking it out. You can observe here, this way we are taking it out. This way we can separate it out from the onion. After separating it out, we are cleaning it with the pure water. That means there should be nothing, any organisms should be not there. Then we are putting it in a saffron solution, because while observing there should be a clarity. And afterwards we are again keeping in the plain water. Whatever the dust things and all is there, that it should be clear. And afterwards we are keeping it on a slide, and with the help of this dropper we are keeping glycerin on it. Put a glass on it, through that there should be no bubble in it. We have to be very careful with it, and whatever the extra glycerin is there, we have to clean with the paper or cotton. Now we can observe this slide in the microscope. After observation you will get this image as we have seen in the picture in your textbook. Exactly same it is there. This is zooming is there. You can observe each and every detail of the same. So you can observe here that the linings are there, that is cell wall. You can observe here nucleus also, and this is cell, the gap which is presented there. Okay, I hope you all have understood. So this onion peeling we have seen. So you have to draw this onion peeling also in your book. So I stop my video over here. Remaining topic we will discuss in next lecture. Thank you.